Okay. All right. Well, thank you, Carrie, for inviting me. Absolutely. Thanks, everyone, for being here. It's actually my first time in North Dakota, so it's a pleasure to be here. I'll be talking a little bit about what we have going on dicamba. I know there was a little discussion earlier of what, either there's yield loss or no yield loss. It's a little controversial subject, but I'm just reporting the data that we have. We can discuss off topic my opinions about that. Okay, so this research started pretty much 3 years ago. In 2019 was the first time we were seeing off-target dicamba damage in Southeast Missouri. If you're familiar with the area, pretty much you have off-target dicamba there everywhere. That's actually the place that they sue Bayer because of that peach farm. So we're very close to the location. So 3 years ago, this one, we started seeing something like this. We were walking plots, and then we would see like the extent check, where you pretty much don't have any symptoms. We would see something like this one, which is a known extent variety, which was completely damaged by dicamba. And then we would have this somewhere in the middle, where you just wouldn't see any typical symptoms from dicamba. We were seeing that across multiple fields in 2019. It was very interesting. And then we went to our progeny rows. So basically, progeny rows, you have biparental populations, parent 1, parent 2. And we were seeing segregation among sister lines. So these side by side, one completely damaged by dicamba, the other one, no symptoms. So for us, it was very interesting. It was a good indication that this could be a trait. So we decided to pursue this research. And it, by accident, it became my PhD project at the time. So just to show how this repeated over years, this is just from last year, our advanced yield trials. You have this, which looks like the extent check, but it's not. You have this one completely damaged by dicamba. And if you look a little further, then pretty much the same thing. So susceptible, tolerant, and so on. And then in our mapping populations, which is one of the components of this research, you can kind of see one of the rows here, no symptoms of dicamba, one in the middle completely hammered by dicamba, and then this one kind of in the middle. So the same observations that we had in 2019 repeated in 2020 and 2021. All right, so what we did about it, I like to call it large scale because it was a lot of work to do all this screening. We looked a little over 10,000 plots over those 3 years. We did all the visual scoring. So as you can kind of see on the scale here on the bottom, we went from 1 to 5. We collected yield for all those lines. We started doing drone because it's a lot of plots to walk and score. And basically we have 3 types of populations in this work. One of the things in our program, we're big on developing, breeding lines and varieties. So we have roughly 550 high yielding breeding lines. Those are all known extent, conventional Roundup Ready. But we did all the assessment on those breeding lines. We have mapping populations, so roughly 480, kind of like I said before, biparental lines, one tolerant parent, one susceptible parent. And then lately, what we have is exotic soybean germplasm. So it's basically a panel of PIs, kind of represents all the genetic diversity that we have available. So if there is differential response in dicamba here, that's a good thing. We can identify what's causing that. All right, so one of the things that we discussed a little before this talk today, we haven't sprayed 1 ounce of dicamba. This is all from our neighbors, and like we discussed earlier, that can go 15, 20 miles away from us. We don't really know where it's coming from. So what we did to at least know that the distribution of dicamba was uniform, we look at the performance of the Roundup Ready check to the extent check. So if you assume that they have the same genetic potential, if there's differential response, that's because of dicamba. So, and we had the same checks across all those fields, and we can kind of develop this type of map. So it's basically a relative performance yield map. And then using what we did here is a Poisson point process. It tells you if that distribution is homogeneous or not. If it's homogeneous, you can assume that this the distribution is the same across the field. So you can kind of see that that distribution was pretty much statistically homogeneous. So for us, it's good. The second thing that we did, and it's the problem of visual scores, we kind of look at how consistent and correlated those scores were across environments and within environments across replications. So in terms of correlation, as you can kind of see on the scale, the lighter colors are low correlation, darker colors are high correlation. And fortunately, all the correlations are dark. So it shows that the scores, although visual, they're pretty consistent across environments and across years. And the other thing that we did is a Cronbach's alpha score. So basically it's a score that it quantifies the consistency. So we did that across the 3 years. It goes from 0 to 1. So we got a 0.95. It shows that the scores were really, really consistent across years. That means that if you have one variety that was tolerant in one environment, if it is high, it was tolerant across all the environments. And it was a lot of environments here. All right, so that's the very controversial part. So just reporting the data. So this is 3 years of yield trials. Like I said before, a little over 10,000 plots, 550 different genetic backgrounds. We did a, mixed linear model, and basically for every increment in that scale from 1 to 5, it reduced about 9% of your yield potential. And this is related to the extent checks in the same field. So to me, it's really interesting because if you think about, especially you have all those specialty projects, if you're growing a non-extent variety, depending on your genetic background, you may lose 10% of yield because of off-target damage, or you may lose 50%. So it's not the same across all the varieties. There's a big genetic component on how those plants are going to respond to dicamba. I think it's pretty obvious, but it was nice to see that maturity definitely has an impact on the response. Later maturity varieties, you have a little bit less yield loss. The window of recovery to dicamba is higher. However, and this is where it's interesting for us, is that the tolerant lines, they perform the same no matter what is your maturity group. So if you look at this plot here, I'm sorry, it's very small, but this first group here is the tolerant. And you can kind of see the yield potential is all up there. There's no difference. They're all in 90, 93%. However, when you go to the susceptible group, you start seeing a big difference among maturity groups. So for us, it's really cool to see this because the tolerance is something besides maturity. So there's more mechanisms besides just your window of recovery, which is good. All right, the second thing that I want to talk about is kind of replacing my eyes and giving the phenotype with a drone. So having a more digital, more precise, accurate way of classifying those lines. So this is a pretty straightforward drone. It's an off-shelf drone, RGB camera. There's no fancy, multispectral camera. It's basically an RGB. And we extracted multiple features that kind of represented what we were seeing on the field. So like we mentioned before, your canopy size is clearly different among a tolerant and a susceptible line. Picture doesn't really do the justice, but your color changes a lot. Intensity of the green changes. So we use those as a feature. And also the texture of the plot is completely different. If you think about it, dicamba tolerant, very smooth. When you have the symptoms of dicamba, it becomes very rough. So we have contrast and entropy representing the texture as a RGB feature. The model is really good when you're differentiating what is tolerant and what is susceptible. So for that end of the spectrum, it really can differentiate what is tolerant and susceptible. In the middle, it's a little gray area. It can kind of go both ways, so your accuracy goes down a little bit. But if you're using that in in a large program and you have to screen thousands and thousands of lines, it really works. It does a good job with that. I also want to mention this because I imagine Carrie was going to be interested. In our exotic germplasm screening, you can kind of see that this is all in the same field, multiple different PIs. So exactly in the same area, you have one PI that doesn't show any symptom from dicamba. You have this one where you barely have a canopy covered, and then somewhere in the middle. And if you walk that field, you're going to see all kinds of responses. So it's really cool to see that you have the genetic diversity available if you want to breed for that. And here's a little closer shot. Tolerant, one of all the typical symptoms from dicamba. So what we can conclude, and then what is next on this project. So first one is that the damage caused by the off-target dicamba is not only cosmetic. Depending on what you're growing, depending on your genetic background, you're going to observe or not observe yield losses. In our case, when you combine that many different genotypes, it does cause yield loss. We just got, actually 2 days ago, this paper has been accepted for publication. Hopefully it will be out in a couple of weeks. The other thing to me that it's interesting and I want to share with you guys is that there's genetic diversity. So if you're interested in growing something else than Xtend, you may have hope to, protect yourself from that. A very simple drone, RGB derived features can pretty well distinguish what is tolerant and susceptible. This has also been accepted this week. So those two papers. So this is coming out in remote sensing very soon. And then what we have going on is the dose response studies. We really need to know how well resistance can handle the dicamba. So we're playing with different doses in control environments and seeing how well that can work. We have all the genomic studies going on. We did some preliminary GWAS. Yes, there are peaks. Yes, this is genetic. We're going through the process of really identifying what are the genes and if we can make that a little more precise than just reporting QTLs. And lastly, again, one of the big parts of our program is the development and release of cultivars. So we have many populations derived from tolerant lines, and those hopefully will be out soon. And with that, thank you very much. If there's any question, I don't know if we have time, but I just want to show this in my last slide. This is one of our most tolerant lines developed from this project. And this is just one regular Asgrow check. And you can kind of see if you draw a line here in the middle, it's pretty much day and night difference. So knowing that this differentiation is not only cosmetic, there's actually yield benefit from this trait. It's very exciting to me. So that's it. Thank you very much.
So now it's lunchtime. So I think that if you have questions for the students, you can bother them when they're eating.